So today I'm welcoming Dr. Claire Carvell from the UKCEH for Ecology and Hydrology. And Claire is going to talk to us about 20 years of research at UKCEH and how they've been monitoring, modelling and mitigating UK insect declines, including a little bit of a focus on the wonderful pollinator monitoring scheme that's been set up. So there'll be information there about how you can get involved yourself or get groups of volunteers involved there as well. So Claire, over to you.
Thank you so much, Kieran. Hello, everyone. And yeah, I wanted to just start by thanking Kieran and his team for all the really great work you're doing to, provide this platform for us all to share information and knowledge. I actually had no idea quite how extensive the platform was until your introductions this morning. So it's really great. Thank you. So yeah, in this talk, I'm going to introduce UKCEH, for those of you who might not be familiar, who we are and a little bit of what we do. And then I'm going to talk about how our research is contributing to really trying to better understand the status of UK insects through modelling existing data and also monitoring to collect new data. And then I'll finish off with some examples of where our research has looked at the impacts of various land management options to mitigate against these declines. And all the time I want us to keep in mind some of these inherent features of insects and their life cycles, which make them quite challenging systems to work with. But of course, that's what makes them fascinating and fun too, and that's probably why we're all here. So UKCEH, we are, as of 2019, we've been an independent not-for-profit research institute. We specialise in integrated environmental monitoring and problem solving across all environments, land, water and air. We have more than 500 scientists now based across 4 sites in the UK. We also have a new office in Africa, and our research is very much global in scope. But to give you a little timeline and some elements of UKCEH, which hopefully some of you might be a bit more familiar with, forgive me, that's shot ahead there. So of course we had back in the 1960s and 1964, the Biological Record Centre was established as this sort of national focus in the UK for terrestrial and freshwater species recording, and BRC now celebrates its 60th year this year. In the 1970s, we saw the launch of sort of large-scale mapping and using satellite-based monitoring combined with ground surveys like the Countryside Survey to start to get a better understanding of, for example, how hedgerows and plant species were changing across the countryside, again, combined with data from the BRC. And of course, we saw the setup of the first transits as part of the UK Butterfly Monitoring Scheme in the mid-1970s. Coming to the 1990s, we began working a lot more closely on sort of applied research, working with farmers and having field trials, particularly linked with the agri-environment schemes, which I'll refer to a little bit later. In 2008, the BRC and our research group relocated from Monkswood. We can see some of the group at the bottom there to Wallingford in South Oxfordshire, where we are based now. And then we've had various other initiatives, particularly focusing on insects. For example, the Insect Pollinators Initiative. I've been lucky enough to be working on some of the molecular advances that have allowed us to understand more mechanistic elements of change in pollinators over time. We've worked a bit on impacts of pesticides on pollinators, and now we are focusing a lot more on sort of whole systems approaches through our programmes, looking at sustainable agriculture, routes to net zero, again, focusing not forgetting the role of biodiversity in our farming systems, and programmes and schemes like the UK Pollinator Monitoring Scheme, which I'd also like to come on to a bit later. So through time, the Biological Record Centre has obviously been embracing some of these new approaches and technologies, but really I think it's retained its kind of core focus and now is supporting, I believe, 87 species recording schemes, collecting unstructured records, 8 monitoring schemes, collecting structured data, repeated surveys over time, and a further sort of 10 other study groups. So again, I'm very sort of privileged to be working with colleagues from BRC, and there's lots to celebrate, again, looking on the BRC website and indeed, obviously, the outputs from various schemes themselves. So there's obviously been a large number of studies reporting declines in insects published in recent years and recent decades. But actually, for most insect species and in most countries, there simply are no long-term data to allow us to really adequately measure trends over time. And so this narrative on insect decline has become quite nuanced. And there's in particular been quite a few calls for more structured, robust long-term data to allow us to better understand changes over time. And also for these analyses to kind of properly consider the inherent features of insects and their ecologies, which make them challenging systems to work with. For example, their short life cycles, their responses to, strong responses often to annual extremes in weather and so on. So if we just start by looking at one of our best recorded insect groups in the UK, so with data on Britain's up to 900 species of larger moths. And so many of you will be familiar with, again, the reporting of data from the Rothamsted Light Trap Network. This is just a snapshot from the latest state of Britain's largest moths. So if we look at the sort of average annual population index, which is plotted on the left, this is showing with a sort of 95% confidence interval in the grey, but a fairly clear linear trend, which is suggesting up to a 33% decline in total abundance of moths over that period. Within that trend, there are 427 species included, so a large number of moth species still not being included despite that really good structured monitoring over time. And of course, we see that some species have decreased, 41 species have decreased, and a few small proportion of species have increased, with quite a number of species apparently remaining relatively stable over time or not showing a significant change. A slightly different version of that same data set was plotted, and interestingly, it kind of shows us how if we plot abundance without the linear trend, we see quite a number within that time series of short-term increases that you can see in the dark blue in that plot on the bottom left, but also decreases in orange. And that kind of, again, tells us that it's really important that we give these monitoring schemes time to detect longer-term trends over a longer-term period because of these short-term fluctuations within that time frame. Looking at moth distribution, so species records from the National Moth Recording Scheme are in slight contrast. These are actually showing that moths, on average, could be increasing by up to 9% in this last 50-year period. They've got trends for 511 species, so again, still by far not the full complement of moth species in the UK. And again, a mix of species decreasing and increasing, and some not showing significant trends. Overall, more species happen to increase than decrease in this particular version of the moth dataset. And again, researchers who've looked more closely into this are often finding there are quite complex patterns when we're trying to disentangle the drivers behind those trends. Touching briefly on the equivalent report for UK butterflies, again, we see a mixed picture for the butterflies, but with a much larger proportion of the UK species being modelled. And again, due to this large extent of recording effort through the Butterfly Monitoring Scheme and the Butterflies for the New Millennium records. But in general, with the butterflies, we see more decreases than increases. But of course, for most of the other insect groups, globally and in the UK, we've really got no sort of structured, repeated monitoring scheme, aside perhaps from the bumblebees, for which we've had the bee walk transects undertaken for the last 10 or 12 years. But this has meant that we've been using models similar to those that were looking at the changes in moth distribution that I just presented to try and understand these trends in species occupancy using the records that we do have that have been submitted through the Biological Records Centre from the recording schemes and societies. So I'm just going to give a few examples from the bees and hoverflies. And of course, we need to recognise the huge amount of expertise and time that is provided by BWAS in this case, Bees, Wasps and Ants Recording Society and the Hoverfly Recording Scheme, but all the other equivalent scheme organisers and members that are gathering and curating species records, allowing us to look into them more closely. But again, because these data are quite sort of noisy and they contain various forms of bias in where and when the species records were submitted, they do require some fairly high-end analytical modelling methods to try and account for that bias. And in this case, we are using what we call hierarchical Bayesian occupancy models to try and help account for some of these ad hoc nature of the records that are behind these occupancy data sets. I think there might be some links coming forward to the work of Nick Isaac, where Nick's written a couple of really nice blogs on, again, sort of dealing with noisy data. But here are some examples of how we've applied these species occupancy models, looking at change in occupancy of 1 kilometre squares over time for the bees and hoverflies. And this plot simply shows you a separate point for each species trend with the sort of 95% confidence interval shown by that line. And we can classify these species as to whether they're increasing or decreasing based on whether that line spans 0. So you've got a number of points below the line that are indicating decreases in species, with the bees in orange and the hoverflies in blue, and a number of points above the line that are indicating increases since 1980. And of course, we can delve into that and see what species are sitting in different parts of this plot. So as an example of an increasing species, the ashy mining bee, Andrena cineraria, seems to have shown up to a 5-fold increase since 1980 in its occupancy. Another one of the Andrena species, in contrast, the Andrena bicolor, is perhaps a more common and widespread species, but seems to have been showing a decline, so up to 42% decline in occupancy. And we can combine all of these individual species trends to produce what we call a composite trend, to try and describe the sort of average change in distribution across groups of species. Here, this is for all of the bees and hoverflies, for which we had sufficient records to run the model. And this is, again, taken from actual data behind the UK biodiversity indicators, which now have a pollinator indicator included. So in this case, we've got a line that represents average change in occupancy across 377 bee and hoverfly species. And importantly, not only is that showing overall that there seems to be a decline in species distribution by up to 30% in that time frame, but of course, within that trend lies quite a lot of complexity. Some species are increasing and many species are decreasing. And again, we know we've not even covered a full proportion of the complement of bees and hoverflies in the UK, with there being a total of about 522 species. But you'll see the sort of plots on the right, the sort of proportional bar chart that shows, interestingly, we can look at both the long-term trend, but we can also start to ask, is there any difference in what's been happening over the short term? And there is an indication that the severity of declines on average may be tailing off across some species. We've got a levelling off of the proportion of decreasing and increasing species. And again, these are the sorts of trends and results that you'll see coming into both the Red List assessments and the State of Nature report that Kieran referred to this morning. But I mentioned again, we're still dealing again with records that are collected in a fairly ad hoc fashion, and obviously will contain some forms of bias, whether that be spatial bias according to where recorder activity is happening. People might tend to go to the nicer sites, and perhaps less interesting arable areas may be under-recorded. And of course, that pattern of spatial recording may be changing over time according to where potentially the most active recorders are based. We may also then see taxonomic bias coming in from the release of atlases or guides, and again, the kind of species that are of particular interest in a particular decade or period of time. So recently, our colleague Rob Boyd and others working in BRC have been kind of further exploring different ways to try and assess and understand these biases, particularly in ad hoc occupancy records, or indeed any other studies of temporal trends in species within the sort of ecological research area. And what this has involved has been taking either individual species or groups of species and looking visually at how we can, again, assess and visualize these kinds of bias from various different angles. So this is an example here for the Plecoptera or the stoneflies using data from the riverfly recording schemes. And what it's showing us is, if you look at the number of records on the top left, starting from 1970, we've had very few records, suddenly starting to peak during the 1980s with a huge amount of recording activity, which then has actually really quite significantly died off following 2000, coming up to 2010, and in the latest sort of decade to 15 years of that time period. The graphs below is another sort of output from this risk of bias assessment, which shows you just how clustered the spatial recording of the stoneflies has been, very highly localized into the northwest of England. And then, of course, that's going to have implications for the shape of the occupancy trend that results from that data. So what we see, there's a couple of species of stonefly in an example here, showing a huge amount of variation in those early and late time periods. And then when you amalgamate to produce a composite trend across the stoneflies, we've got really this U shape with a huge amount of variation at the start and end of those periods. And one recommendation from understanding a bit more about these biases or the risks they might pose to our interpretation of the trends is to think about potentially maybe we should only be assessing trends during that core period of recording in the middle of the U-shape and restricting the sort of temporal inference of our analysis or our conclusions just to that period. My colleague Gary Powney's been taking this work forward under the Druid project, which is a NERC-funded project collaborating with Reading and University of Leeds and Rothamsted. It stands for Drivers and Repercussions of UK Insect Declines. And what Gary's done is applied this kind of risk of bias framework across a huge number of insect species and groups. So he's applied the risk of bias assessments across 19 different insect groups and produced one of the outputs being this really fantastic Our Shiny app, which is kind of a really nice tool, like a resource for recorders or for those interpreting the data. To look, if you can see on the left, you can look both at the sort of taxonomic group trend summary. And again, you can stroll down across all these different insect groups that have been included with their recording scheme cooperation. The species trends are then individually displayed through a sort of click system. And importantly, underneath that, with each one, you see the full risk of bias assessment. So again, to understand where those records are coming from and how widely they might represent the actual real distribution or population range of the species in question. Gary's been working behind the scenes to kind of pull together all this information, and we have some really interesting sort of summarized risk scores across these 19 different taxon groups. We've done some fantastic expert assessment with recording scheme organisers, and we're really excited to be putting that together in a paper, which I'm going to say watch this space to see how that kind of comes out overall. And of course, that will be presented alongside the resulting trends from sort of updated models across all of those different species. And the next step with the Druid insect datasets is obviously to understand, well, why are we seeing these changes? So we'll be modelling them against a number of driver datasets and have already started doing that. But another really interesting paper I wanted to just highlight from our colleagues at University of Reading, Tom Oliver and Luke Evans, they've been looking kind of higher up the food chain to see what are the impacts of changes in insects on potentially birds and bats who are using these insects as key food items. So the Reading team produced this really nice paper. It's just a really sort of a single figure from it here, which is showing some quite interesting links between blue tit populations and moths. So they've used, again, the Rothamsted Insect Survey moth data, and it suggests that in years that have had increased population growth for blue tits on the left-hand axis, correlated strongly with kind of high years for moth abundance. So I'm going to take us back now to talk to you about the UK Pollinator Monitoring Scheme. So again, given the lack of structured systematic records for so many of our insects, and coinciding with the launch of the national pollinator strategies for England, Scotland and Wales, we were able to develop what is effectively the kind of probably the first scheme in the world that's generating systematic repeated data across this many flower visiting insect groups. So we're looking at abundance of bees, hoverflies, and many, many other flower visiting insects at a national scale. POMS runs 2 large-scale surveys, and again, it involves volunteers, citizen science and professionals, and a combination. And we're measuring change across the UK and have been doing so since the scheme started in 2017. The 2 surveys, which I'll talk a bit more about in a moment, are the flower insect timed counts or FIT counts, and a more systematic survey of 1 kilometre squares across the UK. POMS very much underpins the evidence base behind the national pollinator strategies for England, Wales, Scotland and Northern Ireland. Of course, we've got the All-Ireland Pollinator Plan as well. And importantly, we're making our data fully available at full resolution. That, again, some of that's already published on the Environmental Information Data Centre through the POMS website. But also we're working hard to produce metrics and indicators which will hopefully contribute potentially in a couple of years' time, 3 or 4 years' time, towards some of the UK's environmental targets, which are already becoming quite well developed. So POMS, the FIT count, for any of you who haven't become familiar with it already, is a count of abundance of flower visiting insects. And importantly, it's giving us really interesting information about plant-pollinator interactions. Can be conducted across a variety of habitats and places, so gardens and countryside. Effectively, you can do a FIT count anywhere in warm, dry weather from April to September. It's a count of all the insects that land on a patch of target flowers. So the flowers on which you're focusing need to be specified. And we have a list of 14 target flowers that are widespread and abundant that we're really hoping to focus on. But in fact, a fit count can be done on any flower that you choose, as long as you're able to send us a picture and tell us what it is. Importantly, on the fit counts, the insects are identified to group level rather than species level. So again, it makes it a really accessible and engaging activity to kind of interest people in looking at insects visiting flowers. We have had a fantastic uptake of counts. So this is a plot of the cumulative number of counts over the different months of the season in each year. You can see where we started in 2017, down with the blue line was sort of like a pilot year, and year on year we've increased in the number of counts submitted. In 2021, which is the sort of light blue line in the middle there, we saw a really nice upturn when we released the Fit Count app. Yeah, and you can see that just in the top right there. Just where we are this year for 2024, we're not doing too badly, but in terms of receiving counts, I think that pink line in the middle there is slightly down, potentially due again to the poor weather and people just not being able to get out and undertake fit counts. But we'd really encourage you to keep counting, keep submitting information, and of course, a count of 0 is just as valid as a count where you're observing lots of insects. One of the ways in which we're able to visualize the results from the fit counts are these really nice, what we call them the kind of donuts, but this gives you a picture of the typical community of insects that's been visiting flowers on the submitted fit counts in different habitats in this particular example. As you can see, gardens, all of the fit counts in gardens up to, I think this is 2022 data. You can see bumblebees and honeybees are a big component of the community there. If we look at all the counts coming in from agricultural habitats, again, you can see, as you would expect, the insect community is quite different. Lots of small insects and other non-syrphid flies. And importantly, being able to sort of look at how these communities are changing over time is something that we're keen to be doing as the data set develops. It's also been a really nice opportunity to engage with other partners. So here's us doing some workshops with the RHS, looking into their plans for pollinators list. And as I said, a really wonderful opportunity to get data on flower insect interactions. These are just a couple of lovely photos from one of our volunteers, Kay. And just at this point to note that we're actually running the fit count in 9 different countries now. So we have the app operating in South America and various different countries across Europe. So with luck, we may start to have a really nice sort of global data set that is using this structured recording method of insects on flowers. We've also released a new feature with the fit counts, which is this sort of local projects feature, whereby as the scheme was, before this feature was introduced, anyone could upload and observe their own counts. You can go on the website and get an output of your own fit counts at any time. But this feature allows projects, local groups, community groups or researcher groups to actually bundle together a set of counts by tagging their project in the app. And please get in touch with us if you think you've got a project or you'd like to add your organisation or your project to this list. And we can tell you how to go about doing that and how to access the data. POMS, importantly, also runs what we call the 1 kilometre square surveys. So this is a more detailed kind of whole day systematic survey of pollinators and other insects across a set of 95 1 kilometre squares across the UK. I'll show you a map in a moment. Importantly, it's designed to detect broad UK level changes in abundance, in species richness. We're able to look at some species, but not species abundance of all species, because the way they're sampled, and in species distribution. It also allows us to potentially integrate these data with the occupancy models and the records coming from the recording schemes. The sites are stratified, so half of them are in mainly agricultural landscapes and roughly half are in mainly semi-natural landscapes, but those sites are chosen in proportion to each of the countries. So we've got more semi-natural sites in Wales and Scotland than we have in England, for example, and again, a mix of upland and lowland environments. So again, here's the map of squares. If you look on the POMS website, you'd see that those that are in blue are adopted by volunteers. Volunteers are introduced by one of our fantastic survey mentors who are based in each of the countries. So they have an initial visit where they're given training and familiarity with the access points, taking the sampling and all the equipment's provided. But we do have some sites that are still not adopted by volunteers. These sites are still being surveyed, so the team again visits these sites on 4 occasions a year. and so allowing us hopefully to still continue getting data. But yeah, please again get in touch if you're keen to get involved. And there are some lovely descriptions of some of the squares. This is one of our squares in Scotland. Miranda Bain is our survey mentor, one of our survey mentors in Scotland, and again working with one of our new volunteers last year, Enid, who I think hopefully enjoyed her first year of surveys. We've collected a huge amount of data across this network of 95 sites. And what I didn't specifically mention was the main approach that we're using here is to undertake pan trapping across 5 different locations on the site. And it's a sort of full day pan trapping session of 6 hours per site per day. So it's a sort of 1 person per day survey. And of course, we typically generate quite a number of different insects represented in these pan traps when they're left out for 6 hours. This pie chart shows you the typical composition of a POMS pan trap across all the sites. And again, as we fully expected, the bees and the hoverflies, which are the sort of target species of the scheme, are not actually forming a large proportion of that community that we're catching. But we do analyse and identify all of the bees and hoverflies to species level. So we've just got a picture on the left of Nigel, Liam and Richard and some of the taxonomy team in the CEH lab comparing specimens and getting ready to take those away for ID. Overall, we've recorded, I think, up to at least 250 bee and hoverfly species since 2017 with the POMS Pan Trap network. And we've had some quite interesting finds and records, often extending the range of several species beyond the kind of current known ranges. So here's a few species from some of the earlier surveys, that records of interest that have been generated within the pan traps. And our latest annual report from POMS gives you some stories about some of the other interesting species we've found, particularly there's examples of the dark copperback hoverfly, yellow club glasswing hoverfly and the tormentor mining bee. So do have a look at the annual report if you want to find out where we're recording some of these more interesting species. Of course, we're aiming overall to be able to analyse the POMS data to detect changes in abundance or species richness of these pollinators over time. And one of the things we're also trying to do with the sort of modelling of the data is to understand some of the factors that might be affecting the insects that we're recording. And to that end, the survey involves collecting various bits of other information to tell us about the sort of context of your survey. So I mentioned we just published our latest annual report. This has got a 6-year analysis. There's a bit of a time lag because of the amount of processing and the taxonomy that needs to be done on the specimens that we collect. But what we do with these models is we're able to take, as I say, into account some of these environmental factors that are being recorded at the time of the survey. So that includes, for the fit counts on the left, the number of flowers in the 50 by 50 centimetre patch that you count for a fit count, the context of the patch, whether it's isolated or within a patch of larger flowers, of course, the year and the month of the survey and the date of the year, and also some variables that describe the weather at the time of the survey, like wind speed and the proportion of sunshine. And we apply a similar kind of set of variables behind the models for the pan trap data as well to try and iron out some of the variation that's being produced. So these are the sort of plots that you'll see emerging in the POMS report. And very careful to kind of show this isn't necessarily a trend, it's only a sort of 5 to 6 year time series. It's not really enough to start talking about long-term trends, but it gives us an idea about how this is the fit count data set. So on the left, these are all the counts collected by what we call the public fit counts that are conducted anywhere, large number of counts, often in gardens. And the set of fit counts on the plot on the right are those that are conducted within our 1 kilometre square network. And these act as a really nice kind of check for what's happening in those squares against what's happening in the sort of wider fit count data set. Again, you can see the sort of proportions of the different insect groups that we record and generally fluctuation over time, year on year. But what we have seen is a bit of a downturn in the number of hoverflies and perhaps all insects in total in 2022, that was probably in response to that very, very hot, dry weather. The equivalent plot for the pan trap surveys. So again, this is in the latest annual report. Again, these are modelled counts or predicted counts on the left-hand side, and it's shown on a log scale. I should have said that before. So on the left, we've got the abundance of insects in the pan traps. Again, you can see there the bumblebees, solitary bees, hoverflies, and then total number of insects in the top line. And on the right, this is the species richness, so the number of species per pan trap station for the bees and the hoverflies. And again, there's an indication of a downturn in 2022 that we think was particularly related to the hoverflies being recorded in fewer numbers and species because of that very, very hot weather. In relation to what I was saying about the insects that we're sampling in these pan traps, we do in the POMS lab here at UKCEH do a sort of crude count of all the other insects that are sampled in each pan trap. So this is where the sort of brown line comes from in the top left plot here, and the grey line for the other flies, for example. We've done a total count of all of those insects in the pan trap, and then we've put all those insects back into the sample tube for archiving, whilst the bees and hoverflies have been individually labelled and tubed separately for species ID. But what that's meant is that we've got this fabulous archive, and we've been very lucky in the last two to 4 years to be able to start exploring other ways of understanding what insects we've been sampling in these other non-target groups, so the non-bees and the non-hoverflies. So I just wanted to briefly tell you about the latest project that's a collaboration between us and the Wellcome Sanger Institute in Cambridge. Again, this has allowed us to barcode nearly 10,000 insects from a total of 31 POMS survey visits across 4 sites and 6 years of the time series. It's part of the BioScan UK project, which is led by the Sanger Institute, and it literally allows us to individually barcode every single insect that has been sampled. I'm going to give you a very, very quick snapshot of some of the results. What's interesting about these is they're not just allowing us to look at species. Indeed, we know that we can't necessarily match every single DNA sequence to a species yet. And again, this is a problem with sort of libraries and the taxonomy that we're looking to further explore. But the plot on the left just shows you the average number of sort of genetically distinct species. So think of it as a kind of DNA richness, and how that's changing over the years in these samples, and how it differs across the POMS sites that we included in this study. So looking at the non-syrphid diptera, this site in pink at the bottom, super interesting. It's a very wooded site, deciduous woodland near Goring, and it's produced much lower numbers of the non-syrphid diptera than at the other sites. So just really showing us that the communities are quite distinct at these different sites. And again, the patterns are different if we look across all the other species, excluding the non-syrphid diptera, which are clearly dominating the samples quite heavily. So this is a sort of watch this space. We're really excited about how this work on barcoding of all the other POMS pan trap species is going to develop. I'm going to try and speed up a bit, Kieran, because I can see the time. But just to say that, again, undertaking this DNA work gives us a really nice opportunity to feed back to the volunteers. Again, no insects are going to waste, as it were. Everything that's been sampled on these sites ideally has the opportunity for us to find out more and increase our knowledge about what's happening. So each of the volunteers basically gets a species list of everything that's been found on their site each year. So just moving on to the sort of mitigation bit of the study, I mentioned earlier the national pollinator strategies, and of course we've had many different schemes developing, looking at sort of the agri-environments over the last couple of decades. But in particular, the National Pollinator Strategy for England has had key actions aiming to support pollinators on farmland, initially through the CAP, but now through the environmental land management schemes and other voluntary initiatives. And of course, the outcomes that they're looking for, as published in the strategy, are bigger, better, more diverse and high quality flower-rich habitats, supporting our pollinators. And I think this should say, and other insects as well across the country, but this is particular focus on the pollinator strategy. And together with that, we've got these evidence gathering actions, which is particularly where POMS comes in. But at UKCEH, we've been working for a number of years on this, again, sort of gathering evidence to support Natural England and others implementing the agri-environment schemes. In particular, we've looked at these questions around exactly what kind of habitats are needed, when are they needed on farm, and how can we actually provide these, how can we manage them, how can we establish them to get the best for the value of the land that's being put into potentially these environmental options. So we looked in the early years a lot at differences between a sort of simple nectar flower mix, which I'm sampling here, a mix of mostly agricultural legumes, versus a more diverse perennial wildflower mix. And we did various different experiments looking at establishment of those. And we also did some more national surveys looking across large numbers of sites. So this study led by Richard Pywell looked at 150 field margins under countryside stewardship. And some of the key results, again, it probably won't surprise you to know that if you just looked at the common bumblebee species on the left, of course, we see more species being recorded on these more evidence-based, more diverse, flower-rich field margins, for example, the targeted agri-environment options as opposed to the general options, or a control, which would be represented by a standard field margin with land dropped right to the edge. Then if we look at the rare species that were recorded in this survey, they were only ever recorded on these more evidence-based or targeted management options, again, like the species-rich perennial wildflower mix. So this was quite an important development to help look at developing those options and where they should be targeted, and particularly to benefit some of the rarer species. We've also looked into these questions of how much habitat, and in order to do that, we've had to really sort of adapt our thinking to not just looking at the abundance of bees along field margins, but actually asking around more sort of mechanistic questions about how these habitats are impacting their population size and what are the longer term effects of management interventions across the agricultural landscape. So yeah, that's encouraged us to be looking much more beyond the farm boundary and at a landscape scale, also looking at the effects of the surrounding habitat or the context in which the wildflower strip might be established. And in the case of some of the research I've been involved with, how does it affect the behaviour of the insects involved, so the foraging distances of bumblebees in this case. So yeah, we've done some studies where we've sampled the whole landscape, and in the case of bumblebees, we've had the opportunity to look across the life cycle of the bumblebee colony by doing some interesting genetics. So this is just one of the most interesting studies I think that we've done, or that I've been involved with, that was conducted under the Insect Pollinators Initiative, where we were able to sample DNA from bumblebees across a landscape, and that landscape was structured. It's not a very big photo on the far right, top right side there, but we had various bits of the landscape that were rich in floral resources for bees and other areas of the landscape that were poor in resources. And by genotyping all the bees, we were able to group each species that we'd sampled into nests or colonies or related individuals that had come from the same queen. By then sort of modelling the positions of all of those bees and the families into which they had been grouped by the genetics, we were able to create this incredible map across the landscape of the sort of predicted location of their nests. And what that allowed us to do was to start answering questions about how far the bees might be foraging across the landscape to get food, again, particularly from these different enhancement options across that estate. So again, I'm going to have to move on a little bit quickly here, but we found, for example, that the proportion of flowers in the summer in a given area of land within the radius of the colony, as that proportion increased, the radius or the distance that the bees were having to forage was reduced. And we were able to plot that relationship across a number of species, including Bombus ruderatus, the rarer bumblebee, the long-tongued species that was occurring at that site. We also then were able to look across generations of bumblebees. So this kind of showing you from the queens in 2011 through their daughter workers that summer to mapping the locations of queens that had survived and were emerging in 2012, the following year. And this is what we termed family lineage survival. So this really exciting result that we found from the work was that across 3 species, the more common species in this case, we found that the amount of semi-natural vegetation that was in proximity to the colony was significantly enhancing the probability of this metric of lineage survival, so the probability of seeing queens that were related to colonies from the previous year. And that was a really nice bit of evidence to sort of back up the implementation of these management options on farm. I want to flag another project, which I'm not going to talk about in depth because of the time, but my colleague Jo Staley has led another one of these sort of national level monitoring projects funded by Natural England, working with BTO as well. And they've looked across a number of mobile taxa, so particularly looking at insects and birds and bats. And they were able to survey across a large number of squares in 6 different regions that were graded along a gradient of the amount of agri-environment intervention that they had. So this allowed them to ask, what is the difference between sampling these mobile species in an area that's not surrounded by agri-environment habitat or in a landscape that does have a high proportion of agri-environment habitat. There's a snapshot of the results here, and hopefully there might be a link appearing in the chat if you want to find out more about that project. But they've produced a really nice baseline report. And one of the kind of key headline messages is that habitat quality, again, areas with high floral diversity were positive for butterfly species, other species, bumblebee species, and so on in that study. So just to conclude a few points, again, it's not going to surprise all of you to hear that sort of the top conclusion there, that we know many UK insects are in decline, but some are also increasing, and some or many appear to be remaining stable over time. But of course, many other species remain fairly under-recorded. The trend outputs that we're able to produce are going to be dependent on the methods we use and the models that we apply, and all of these have their advantages and their limitations. But we're beginning to understand a little bit better how we can use different ways to deal with some of those biases within the data sets. We need to recognise the real challenges for implementing large-scale monitoring of insects and other invertebrates, particularly the taxonomy. Again, that's not going to be news to this audience, but bearing in mind that I think the DNA or potentially AI technologies are maybe going to be a part of the solution to this challenge. Sampling methods are all going to differ, but if we can use consistent standardized methods over time and potentially look at how we can combine those standardized structured data sets with the non-standardized occupancy records, we may get a much better picture of what's happening. And we also need to invest, of course, and keep focusing on the recruitment and retention of our volunteers, and invest in those that are providing the information, going out collecting records and providing them with some engaging feedback. We need to recognise again that these populations of insects are going to be highly variable and sensitive to the weather, so making trends and predictions can be difficult, and we definitely need decent kind of 8 to 10 year decent time periods over which to analyse our data sets. And also recognise that despite having a really nice, in the example of POMs, a really nice UK network now, we're probably still going to require continued targeted recording effort for our rarer species, which I think we may hear about more in some of the next talks. Habitat quality is obviously key for the land management schemes and developing new schemes. And again, we need to push that message that season-long resources can be provided at the right scale, and some of our research is helping to contribute to that information. And finally, we want to keep funding our long-term large-scale surveillance schemes, but these, again, need to complement the question-driven research. So all of the applied research, the question-driven studies that are being conducted with the universities, and I think working together is a really exciting space to be in. So thank you very much. Thank you for allowing me a bit more time there, Kieran. And yeah, I just wanted to acknowledge all my collaborators and funders. Thank you.